So, first of all, I want to thank the organizers. Thank you so much for organizing this wonderful meeting. My student told me that everybody starts with apologizing for those that will not understand him because he's not in the same discipline, so I will not apologize. If you don't understand, you need to switch fields to understand this interdisciplinary research. And today I'm going to talk about neurofilaments, which are proteins that we find in the nerve cells. And they are extremely important in terms of mechanical properties for the nerve cells. And we tackle this problem from a biophysicist point of view in order to understand the roles of the different components that building up these filaments and how do they contribute in the overall picture for what this protein are used for. So, first of all, what is the cytoskeleton? It's the skeleton of the cell. And if you go to any biological textbooks, you're going to find out that when they talk about the cytoskeleton, there are chapters and chapters about two different proteins called microtubules. This is a microtubule. It's just a nanotube, just like a carbon nanotubes in a sense, but it's made out of biological material. It's about 25 or 26 nanometers in diameter, hollow cylinders. It has a lot of properties and a lot of functionality. There's another class of protein called actin. You heard it in one of these talks today about how they involved in the motility in the periphery of the cell. And they are extremely important. And then after you read all of these 2 or 3 chapters about the cytoskeletons and this important protein, you found out 2 more paragraphs telling you that there is a third component in the cytoskeleton, and this filament is called intermediate filaments. And the reason it's called with this beautiful name is because it's intermediate in size. And you see that I gave here the crystal structures even of actin, or the building block of actins and microtubules, but for intermediate filaments I don't have this. And the reason that there is none crystal structure for intermediate filament, at least not for all of them. Now, the reason that they are very interesting is that unlike the other components in the skeleton of the cell, these types of proteins are cell-specific, meaning if you go to the eye, if you go to the lung, the muscle, or the nerve cells, you're going to find a different types of molecule. So they are cell-specific. And as we all know, each type of cells is different from the other. So this is one of the most important components that differs the skeleton of one type of cells to the other. So I will not be able to speak about all intermediate filaments in one talk. I'm going to concentrate on one type of filaments which are found in the nerve system, and they are called neurofilaments. And they are not just one type of building block for these filaments. There are 3 different types of building blocks, and we're going to talk about them. Now, why neurofilaments? Why they are even interesting? And I think that, sometimes one word is better, one picture is better than 1,000 words. So if this is a cartoon of a nerve cell, and this is the axon, this is where all informations are transmitted from one nerve to another. If you do a cross-section on it and you look on the mesh inside, it's not water. If you open up a cell, you don't find water. You find some kind of a jello. And this jello inside is you can see it over here. It's kind of a mesh of fibers. And these hollow cylinders is the topics of these huge chapters that we talked about. This is the microtubules. But all of these dots, so just cross-section, so you just see from the top, all of these dots are neurofilaments. These are the actual components that you find in the skeleton of the axon. Here again, you see that they are actually forming some kind of a mesh inside of the axon. And this is a microtubule. You see, it's a thicker cylinder over here that you can find it. So they are very important because they are so abundant in the axon. So we want to understand how do the interactions between them, or what is their roles on the elasticity or the mechanical properties of the axon, because these are the major components there. Okay, so again, I said that they have a lot of influence on the mechanical properties, but they also have direct correlations. I mean, in measurements or studies that they knock down some of these genes that produce this protein, what you find out is that actually the axon diameter is changing. Because again, this is something of the components that build up the middle, the soup inside this axon. So if you knock down this, the protein that produces this neurofilament, you find out the diameter is smaller. If the diameter is smaller, then the resistivity is higher, and then you cannot have transmissions of the electrical signal as efficient as you want. Another thing which is very interesting is that the composition between the building blocks of this filament is changing either after an injury, but also during development. So if you have a very infinite an axon or a nerve cell, it will be composed from a set of subunit protein, and during the development, it will change the composition in order to match up the necessity mechanical stresses that is going to be applied to. So it's very interesting to understand how these compositions differences affect, in the end, the mechanical properties that these gels have been producing. And the last, which is, in my point of view, the most important, or the most interesting to me at least, is that all of the interactions between these neurofilaments are actually driven by an unstructured domain, intrinsically unstructured domains of the protein. Meaning that it's not just covalent bonding between one place to another that will make the interactions in the end. So, and I think that this is one place where, when soft matter physics can actually help in order to understand what are the mechanisms that are responsible for the interactions. Because it's not something very specific. It's not just this I changed to G, and G is matching to this, and so on. Okay, so let's go on and understand how we build up a neurofilament from the beginning. So I said that there are composition made out of 3 different types of protein. The names are very easy to remember. These names are neurofilament low, neurofilament medium, and neurofilament high. And all 3 of these components have some part which is very, very similar for all 3 of them. It has an unstructured, very short head group, and then about 300 amino acids which are forming an alpha helical. And then the difference comes about the tail. The tail is different. The lowest molecular weight is the shortest one, then medium is longer, high is the longest. So it's very easy to remember. Now what I want you to notice, of course, this is how many amino acids, what is the length of the tail of this different protein, but they are very much charged. So if we count how many free electrons we have on each of these tails, we notice that they are extremely negatively charged. Okay? So this is just the building blocks. How do we build up a filament out of that? The way that we are going to do that is each of these 3 subunits can dimer up. And this is because this alpha helical place is very hydrophobic, so it doesn't like water, so they just coil So just through coil interactions, just because of hydrophobic interactions, they repel the water and coiled up. Here we have some kind of a matching that you must have the shortest one, neurofilament low, to be with either with himself, like a homodimer, or with the other longer one, so low with medium or low with high. And if you take these dimers, and after you have a dimer, 2 dimers will again coil in one into another to form a tetramer. And in the end, after 32 of those subunit proteins, they will form a filament, okay, which will be 10 nanometers in diameter. And we'll have all of these unstructured details coming out outside. So it will look like something like a bottle brush. The brushes that you use to clean bottles. So this is how how the sequence is in order to get the neurofilament. And I don't know if you can actually see, not the cartoon, but the very good electron microscope here. Very hard to do, but you can actually see the little hairs coming out of these C terminals, which are not ordered. Okay. Amino acids. So, okay, so this is how many residues do you have in terms of amino acid. Each amino acid is about 3.5 angstrom. So this gives you what is the total length of it. Now it's a polymer, so it's flexible. So the good question is, what is the actually length that they are going to be extended to? And I'm going to give you some answer about that. Okay, so this is how they look like. They are 10 nanometers in diameter, and about between planes, there's about 22 nanometers. So we are actually in the nanometer world, and so this is very suitable for this talk. Because of this rule that I told you before, and I didn't explain why, but this is actually, this is how it works, we cannot make any types of filaments that we would like. We can only make neurofilament low alone, which is just a homopolymers of all of them just made out of low. This is, by the way, what happens when the neuron is very in early stages. Or we can make a copolymers of low with medium, meaning that we have some low and some medium in the polymers, or low and high, all three of them. In a healthy neuron, the molar ratio between low, medium, and high is 7:3 to 2. In a disease state, it's different. Okay? So we want to understand the relation between the composition to the properties this polymers is giving us. Very complex polymer that is giving us. The first study that we did, and maybe you already heard a talk of mine about it, is that we wanted to understand the structure of the hydrogels that these filaments are producing when they are in high density. So this is an AFM image of this hydrogel that we put after we purify the protein and reconstitute them in vitro, meaning outside of the world in glass. And this is how when they react, they form this beautiful nematic liquid crystalline structure. And in order to study what is the size in between two filaments, we use the small angle X-ray scattering techniques that gives us the correlations length in between these two superstructures, these two filaments. Okay? So X-ray gives us the dimensions in between, the average dimension in between these filaments. And the idea was that using this technique, we can vary the surrounding of this gel and learn about the phase diagrams or the properties of this gel and get some actual numbers about the bulk or the collective bulk behavior. And here this study, we just did a study of changing the osmotic pressure on this gel and by measuring the distance in between these filaments, we did something that I was taught by maybe David when I was a student in undergraduate studies, to make a phase diagram of pressure versus volume when we learned about phase transition between liquid and gas. Here we are doing it for a little bit more sophisticated system of a hydrogel with filaments. But again, this is pressure versus distance in a sense. And what we find out is that we have several phases of when the gel is very expanded. And you can see that the average distance in between two cores of a filament is about 80 nanometers. So you can imagine what is the length of the extension of the unstructured domain. And this is very much controlled by changing the composition of the filaments, the compositions of this unstructured domain, but also is very much determined by the amount of salt that you have in the solution, because this is going to be the mechanism in order to screen out electrostatic interactions of this highly charged tails. And by taking a look on this bulk behavior, one could actually compute what is the bulk modulus, or the compressing bulk modulus for these gels, because it's just a derivative of the graph. Okay, so this was a collective bulk behavior. Now the questions that we ask ourselves afterwards, and I'm giving the question and the answer in the same time, is whether on a single filament length scale, again, this is a supramolecule, it can be in length of a few microns and in diameter of about 10 nanometers, whether the side arms can interact one with another. We learned in this collective bulk behavior about the interaction between one filament to its neighbor. But what about the interaction between the same filaments to himself? Can it bend into the nearest plane of tails? And can this be a competing force in the reorganizations in the gel? And the answer for that is yes. And I'm going to prove to you that it is actually matter. And the way that we are going to do that is that we are going to measure the elasticity, how much a filament is elastic, or measuring what is called the persistent length of a neurofilament versus two very important parameters. One parameter is the composition. How much do I have from low? How much do I have from medium? How much do I have from high? Okay? Because that changes the interactions. And the second thing that I'm going to change, as I told you, these tails are very much charged. So I'm going to change the salt in the solution and going to measure what is the effect of changing the electrostatic interactions on the persistent length. How does that imply on the persistent length values? So this is the task. And first of all, I need to explain to those that do not understand or do not know what is a persistent length. A persistent length is just a measure of the elasticity in a sense. So it's how much length do I need to go along the polymer before I lose my memory about the original direction that I was at. If we take this pen, the persistent length is infinite, because if I'm here, I remember that I was pointing this direction. If I'm taking a cooked spaghetti, after about 50 centimeters or 10 centimeters, it will forget the idea that it was directed into one direction and it will be flipped into another. So this is a matter of how much it is elastic or how much it is flexible, the persistent length. For a DNA, for example, the persistent length is about 50 nanometers. Okay? After 50 nanometers, you will see it was pointing in one direction, in any points on the DNA. After 50 nanometers, if you go along the contour, it will start to move to a different direction. Okay, so the next thing that we need to remember is that we have here a polyelectrolyte. Polyelectrolyte is a polymer which is charged, right? So if we go back on polyelectrolyte, this was in a very active field in the 70s, late 70s, and also was revived in the 90s. And there are a lot of theories about that, and nobody actually knows the real answer in the end. But the message that I want to tell you is that if you have charges along a polymer, it makes sense that these charges will repel each other. If they are going to repel each other, the farthest that they can go is that if they are going to be aligned. So they're going to be stretched out. And if you will increase the amount of salt that you have in your solution, you will screen out some of these electrostatic interactions, and it will become more flexible. So in the end, what I want you to remember is that the effective persistent length will be inversely proportional, and I don't care now for this matter what is the power law dependence on the ionic strength of the solution. As more as you have salt in your solution, you will be more flexible. If you don't have a lot of salt in your solution, then you're going to be stretched out. That's the message. Now, we don't have really a polyelectrolyte. We have something that looks like a bubble brush. So again, here, theories came out, and not so long ago. But again, the basic idea that I want you to remember is that if this is just a repelling interactions between these hairs or brushes hair that you have, just because of steric interactions, just because you want to repel your neighbors, as longer as the brushes hair will be, or as tighter that they will be attached to the backbone, they are going to repel each other and going to stretch it out. Right? This is just common logic. We didn't need to wait until 2005 to understand it, but now we have also the formula for that. Okay, so this all makes sense, right? So now let's ask ourselves whether we can measure it with neurofilaments and find this very common sense also applying for this complex protein. So the way that we're going to do that, and this is a really injustice to how much time it actually took, because this little arrow takes about a month in order to make the protein that we need. We purify our protein from bovine spinal cord, and we were able to separate into these three little subunit proteins, and from about 5 cows, you get no more than 1 gram or 1 milligrams of protein that you need in the end. And again, it takes about a month and a lot of dedicated work, but what you can do, this is biology. And the good thing about this protein is once you have them, you don't need to make any effort in order to get the filament back. You denaturate them at some point in the purifications in urea, and after that, you just need to dialyze them out to water with salt, or buffer if you are a biologist or biochemist, and what you get in the end, you get the filament back. So they are self-assembled. And again, the assembly is just relatively rapid and robust because it's mainly driven just by hydrophobic interaction. So they form these filaments automatically no matter what you want, if you want it or not. So, and then after we had this, and I will not get into the business of how we actually was able to make these beautiful images that we did, but we deposit the filaments on a special film that we were able to capture the proteins on the surface and immobilize them in their natural state in a certain conditions with salt and so on. And we just immobilized them, froze them on the surface and this particular technique. And after that, we were able to image them using atomic force microscopy. Now, we want to measure the persistent length, and the idea behind measuring the persistent length is that you need to have a very accurate measurement of the length along the contour of the protein. So there are some techniques that we were implying, but or some theories that we were using in order to calculate what is the persistent length. Here is the LP. This is the persistent length. We used a very old theory about just measuring the correlation between each point along the contour, and then we went to a more extended theory that also took into account the fact that this has finite length, and a very short finite length, by the way, of the contour in respect to the persistent length. And again, both these techniques require to know very accurately what is the contour of each filament that was deposited on the filament on the surface that we measured. So in order to do that, we understood pretty fast in the beginning of this project that we must have a very good accuracy. We want to measure very tiny changes in the persistent length. We must have a very good technique in order to measure this contour. And for that, so this is the goal. You need to just find out for these filaments, graphically displayed here, what is the top of the heights on it, right? So how do you do that? And usually, if we image it with any type of images, we're going to get some kind of a grid, and you will have the filaments on some of the pixel, and then you need to do some kind of filtering, and you will find out the top of the heights. Now, what we remember is that life is not always white and black, and there is also some gray areas. And in particular, the AFM is very good because it gives you the height profile. So we actually use this information in order to make fits to every point along the contour in order to fit it to some kind of a Gaussian, and from that getting a sub-pixel resolution on the topography of the filaments, and from that we can deduce in very high accuracy the contours of the filaments. Now we did that, and this is how scans looks along the top in an automated way, of course. And I think this is the one thing that is a good message to see whether your technique is working or not. After we measure over 2,000 filaments with 2 different AFMs, with 2 different, me and another postdoc in the group, and a lot of AFM tips, the variations in the actual filaments geometry was not modified. And that was a very good confirmation for us that the technique is working. The only thing that was changing, it was the persistent length. So this is just a reminder. The reminder is that low is the shortest one, medium is the highest one, medium is the intermediate, and high is the highest one, and they are all charged, right? And so now the question how that is going to affect the persistent length when we measure it in different composition and different salts. And this is the result. This is the end result that I want you to go back. The result is that everything can happen. In particular, this is for neurofilament low and medium. This is a phase diagram. This is 0% is just low. 40% is the highest that you can get with this combination. And let's go over this very complicated information that we have here. If you just go for low, as you increase the salt, remember, you increase the salt, now the filament see more electrostatic interactions, you will expect it to be more flexible, right? It goes the other way around. If you do it in the other way, if you do it at high molar ratio of the salt, it goes the other way around from just low. If we go at a certain salt, low salt, and we just add up more longer tails, so now we need to repel each other. What happens is, no, it just goes the other way around. If you do it at high salt, it just goes as you expected. And if you do the same experiments, not with low and medium, but just low and high, you get a different picture at all. So what is the message? Everything can happen here. And the message is that the naive picture that we had about polyelectrolyte is not the case for neurofilament. The apparent flexibility that we measure depends on the composition and depends on the salt concentration. And this is because you don't have just repelling interactions that you will find in electrolytes or that you find in brushes which are neutral. You have something that is also attractive. And this attractiveness is dependent on the composition and dependent on the salt concentrations in your solution. And this is exactly what we also saw in the small angle X-ray scattering study, that we saw that we have some attractive interactions that happens at a different salt and in different composition. And the idea behind this interaction is that when I showed you this table, I didn't show you another part of the table, and it's true that they are negatively charged, but there are about 40% which is negatively charged, but there is also 20% that is positively charged. So you can find regions along the side arms which are complementary, and by that they attach one to another, just by ionic salt bridges. And this depends on the composition, because the sequence whether it's matching or not matching will make an effect on the final outcome of the interactions. So this can be explained. So we just modified the persistent length theory in a sense to also include this handshaking between the sidearms. And this depends on the sequence, and this depends on the salt, how much these interactions will be relevant. And this can explain all the phenomena that we see. And since time is running out, I just want to thank those that helped. Joanna Dick was the graduate student that worked with me and did the magical in the lab in producing these filaments. MC Chow is my colleague that was helping me in the AFM, and Erwin Frey and Phil Pinkus from UCSB were help on the theoretical arguments on this work. You can read all of this in these papers. If you have any questions, please come to me.
Time for a short number of questions, please.
What is to show about the elasticity of filaments were already shown on actin filaments in microtubules, and were already shown on filaments of actin and microtubules. So why? And already been proven to be Manning condensation types. So why is it new?
Okay, Manning condensation type is when you take different types of ions, and by that you can induce some kind of an interactions between that. Here we only have monovalent salt, okay? And here we don't have the condensations of salt on top of the polymer. In fact, what we have is the opposite. I mean, Manning condensation goes in just one direction. Here we show that depends on the sequence. We can either make it attractive or repulsive. And this is something that you cannot do with actin and you cannot do with microtubules or any other polymers that don't have these attractive interactions, not coming from multivalent salts, but coming from the salt bridges that you can form in the sequence. So this is the main difference, if you want to say, between those. And before actin and microtubules, it was shown for DNA and so on, but for Manning. But anyway.
Thank you for the presentation. I mean, the change in the ionic strength is really good for understanding the different interaction. But if you look from the physiological point of view, you won't expect to have very high, at least not monovalent ionic strength changes within the cell.
Yeah, but so in about 240 millimolars is about the ionic strengths that you will find in a cell. You will find this gradient in concentrations of the different filaments, different subunit protein during the development. So we use the different salt as a biophysicist in order to understand what is the interactions which is responsible.
That's fine.
And we measure it also in a physiological state in order to see what happens during composition differences that you find in different stages of development or after an injury. So you can understand only what is happening in physiological, but from that you cannot deduce what is the type of the interactions. Now, what happens in reality is that you do have this kind of modulation, not by salt, but via phosphorylation and dephosphorylation, which effectively removes a lot of these negatively charged ions from the system. And this is a little bit more tricky to do it in the controlled way that is done in the cell, but you can easily tune it out, a similar parameter, by changing the salt. So this is why we did it as well.
Last question over there.
In the AFM experiment, can you be sure that when you deposit it on a surface, the surface doesn't alter the persistent length somehow in comparison to, like, solution phase? Or
it's an excellent question, and obviously it's something that you're worried about a lot. I can give you two answers. First of all, we did whatever we can do in order to minimize the surface interactions by using these photoirradiated surfaces that you put the protein on top of them. They are on a liquefy. You can liquefy the surface so they can be running around, and then you just you just solidify the surface, and then they just get stuck like in a murder scene. And so this was the idea why to take this extra effort. But the second thing that I want to give you, that is all of this study, I don't really care if the persistent length is 100 or 95 nanometers. I don't think that anybody really cares. What somebody cares about is whether in comparison from low to low with medium with 10%, so 20% more percent, something changes. And in that sense, I don't think that it really that much matters. Okay? So maybe the actual values are a little bit different here or there because of the surface interactions. But in comparison, in such a elaborated work, I don't think it makes a whole lot of difference.
Okay, we should move on. Let's thank the speaker once again.